Hello, my name is Ali Truckin, and I'm recording this video about the differences between cellular respiration and fermentation for Microbiology 205 and 206 at Arizona State University. Energy in the bonds of organic molecules is transferred to the bonds of ATP. Glucose, which is a great example of an organic molecule, is made up of 7 different CH bonds. Once we start catabolizing the glucose or breaking it down to transfer that energy to a different form, we transfer the energy into a phosphate bond in ATP. Glucose is converted into carbon dioxide once the CH bonds are broken down, and once that happens, the electrons are bonded onto oxygen molecules. This happens during cellular respiration. The main difference between cellular respiration and fermentation is that cellular respiration is aerobic, meaning that it requires the use of oxygen. Fermentation, on the other hand, is anaerobic, and it actually happens because of a lack of oxygen not being present. According to our textbook, during cellular respiration, electrons are taken from glucose to the electron transport chain, or the ETC, which it then donates them to a terminal electron acceptor. The ETC uses the electrons to generate a proton motive force, which is an energy form that is used to make ATP. During fermentation, cells break down the glucose through glycolysis, just like cellular respiration, but instead of oxidizing the glucose, because remember there's no oxygen present during fermentation, it creates pyruvate. And the pyruvate is used as a terminal electron acceptor. Unlike respiration, however, fermentation does not use the TCA cycle. It only uses key components of that cycle to produce certain precursor metabolites. During fermentation, glucose is only partially oxidized, and it produces a lot less ATP than cellular respiration. So cellular respiration is kind of like the go-to way to catabolize glucose into ATP because it produces so much more. Thank you.